Hello and welcome everyone for our webinar today, Natural Language Processing, Rare Disease and Precision Medicine. I will be handing over to Jane now, who will introduce the speakers today.
Hi, good morning, good afternoon, and welcome to our webinar. My name is Jane Reid, and I'm Director of Life Sciences at Lingomatics. And I'm going to be joined by two colleagues, Liz Marshall, Associate Director of Clinical Analytics at Lingomatics, and Sri Devi Murthy, Krishnamurthy, Application Scientist at Lingomatics. So just to give you a quick overview of what we're going to be talking about. So the format of the webinar, Introduction to Lingomatics Natural Language Processing-based Text Mining, particularly talking about where it can be applied for rare disease and genotype-phenotype information. So briefly, Liz and I are going to talk about what is natural language processing-based text mining and how it can create and integrate relevant structured data from unstructured text. Then we're going to talk through some case studies, again, mixing that healthcare and pharma focus. And then Sri is going to give us a demo. And as you can see, we're very happy to take questions. Please use the webinar Q&A form during the webinar, and we'll address these at the end. So first of all, a little bit about Lingomatics. As you probably know, Lingomatics is an IQVIA company, part of the overall IQVIA brand, but very much operating as an independent company. We're headquartered in Cambridge, UK, and have our US headquarters in Marlborough, just outside Boston. Lingomatics provides software and consulting which we deliver based on enterprise and cloud solutions. Our core platform is a natural language processing, or NLP-based text analytics platform. And what NLP is really about is extracting and synthesizing knowledge from text, going way beyond keyword search. The software is out of the box and ready to use straight away. So we focused on solving problems for life science and healthcare organizations, working with 19 of the global top 20 pharma companies, as well as lots of smaller life science companies. We also work with healthcare, payer and provider organizations, and with academic medical centers. We also work with governments such as the US government, FDA, and in fact NASA. We're very proud that NASA is using our Lingomatics software platform to mine astronaut data. So the key part that where NLP brings value is being able to extract information from text, not just the concepts, but also the relationships. So being able to extract concepts and the relationship between them, taking that information to synthesize knowledge using a unique and powerful blend of methods. So going into that in a little more detail, when we think about keyword search, everyone's very familiar with keyword search. But in essence, it's limited, it's resource intensive, it's not systematic, and it's not comprehensive. So when we're thinking about having to answer questions, in normal life, if you're thinking about where to go on holiday, keyword search, putting a search term into Google works really well. But when we're thinking about really answering questions across life science and healthcare, then you really want to get not just a few answers, you want to be able to see the whole landscape, the full picture. So in this little schematic, I'm showing a search for breast cancer. That could be Google Scholar, could be PubMed, whatever. I've typed in a couple of words and the search engine goes away and finds all the documents you can see here below that have the right keywords in it. That's great. But then it's brought back a set of documents. This could be 10 documents, but it could be hundreds or thousands. And bringing back documents means it's then up to you to then read through each document and decide whether it's relevant and extract the information you need. So if you're looking for biomarkers for breast cancer or genes associated with breast cancer, that's a really tough problem. And that's really what NLP can solve. In essence, natural language processing is a powerful data transformation tool, enabling you to move from document-written to data-driven decision support. In essence, what NLP does is take a set of documents, kind of shown here schematically on the left-hand side, unstructured text and using a range of methodologies. So here at the bottom, natural language processing for the linguistics, ontologies for the vocabularies that are important in life science and healthcare, and then statistical methods, machine learning, chemical understanding, regex, and so forth. It can then look through text and extract structured data using powerful queries. Now, once you've generated data in a structured form, that's really valuable. You can use it to drive analytics and outcomes. You can look at it directly, plug it into analytics tools, predictive models, visualization tools, or feed data warehouses, data pipelines, and so forth. In this particular case, in the snippet in the center here, this is a relatively simple example of extracting ejection fraction data. Ejection fraction is a measure of heart health from patient notes. You'll see on the right-hand side, you have the snippets of text with yellow highlights for where the machine has found ejection fraction, blue highlights for where we found the various numerics, and then pink highlights for where we found particular bits of context. And we've been able to find them in the snippets of text and then structure into the columns on the left. So you can see we've got all the different values for ejection fraction, and now where we've been able to find context around estimated or calculated. Again, that's been structured. So again, structuring the data in order to feed, to drive your analytics and your outcomes. As I said, this is just a simple example, and the results can be much more sophisticated than that. I'm going to hand over to Liz for a little bit more about natural language processing.
Thank you, Jane. So natural language processing. Let's start with what it is not. It's not a keyword search or speech to text. What it is the ability to find linguistic patterns from unstructured data, aka free text. For example, in medical records, it is the documentation that a clinician does to ensure that they are thoroughly recording every aspect of the encounter, like in this instance, a phenotypic abnormality. In life sciences, an example may be capturing all the articles that have the numerous ways of documenting a gene mutation. So NLP allows the ability to capture particular insights and terms within the documentation. It allows capturing simple concepts such as acronyms, synonyms, or as well as our platform can capture misspelled words. This gives us the ability to capture such concepts and normalize them as one word or a phrase. Like the example COPD. It can easily be identified as chronic pulmonary disease. On the other hand, we can do complex concepts such as sentiment in something like, say, a customer survey. Context is very important, especially in medicine. Is a disease mentioned in personal history, family history, or perhaps it is ruled out completely? So all of these things are important in characterizing the documentation and making sure we extract the right thing. This may also even involve looking at the document regions and making use of those to navigate to the right information. So when you drill down to look at an individual patient, you can use NLP to extract key insights from text to fill in the missing pieces, to provide a complete picture of the patient or even the population as a whole. So let's take a look at our fictional patient, George. George came into the clinic with shortness of breath. He recently had a long-standing viral infection from which he is having a difficult time recovering from. So his past medical history also states he was recently hospitalized with a DVT. As we dig deeper, we find that he is also lately complaining of nosebleeds, stomach aches, and has been noticing some rather large bruising from very little impact. But George hasn't felt up to addressing medical issues. His wife recently passed away, and he now lives alone. So there are many things we must take into consideration. What is causing the shortness of breath and all the other issues? Could the virus have triggered some type of immune response? Does George now have a rare disease related to clotting? We will need to rule out many diseases, even consider ones that are like acquired hemophilia. As you can see, we don't know about the full patient picture until we dig further into the unstructured data. And from there, we can decide on the appropriate testing. But we also need to do all of this rather quickly. Like in this example, it could be life-threatening. So we talk about rare diseases all the time, but exactly how rare is rare? To do that, we need to turn to the numbers. So we obtain these from sources such as the National Institute of Health and the World Health Organization. While individual diseases may be rare, the total population with a rare disease is actually quite large. An estimated 400 million people worldwide have a rare disease, not a trivial number. So about 30 million in Europe and 25 million in the US. How many rare diseases are there? Well, around 7,000 is a safe estimate, but the numbers do vary a bit depending on the source. So many people think you will know when a baby is born or shortly thereafter if they have a rare disease, which is true in many cases. The majority of rare diseases, approximately 70%, do actually appear in childhood, but there are cases where a rare disease will develop when you are an adult, like George. He had that viral infection that triggered what we later found was a rare disease. But good news. So luckily we caught it in time and our fictional patient, George, is recovering well. Jane, on to you.
Thanks for that, Liz. So as Liz said, there are a big problem with rare diseases, and it's becoming an increasing focus across pharma and healthcare. So of those 7,000 that Liz mentioned, only about 5% actually have a licensed treatment. So there's a very big unmet need, as shown in the graphic in the bottom left-hand corner. So when we're thinking about whether in healthcare being able to make sure you've diagnosed the right patient, understood what their rare disease may be, and therefore if there is a therapeutic treatment available, or when you're thinking about drug discovery and development, thinking about how you could find information around to assist the pipeline of developing rare diseases, there is information out there, but it's very sparse. It's often buried in unstructured text in different data sources, so electronic medical records, scientific literature, and so forth. And it can come in different formats and different vocabs and so forth. So that's all very challenging. Our natural language processing can provide a benefit here. It can bring rapid access to gene disease landscapes, gene mutation associations from that unstructured text. And it enables you to collate the sort of annotations that can be really valuable to build that landscape of information for rare disease, looking at diseases and pathways, phenotypes and mutations, expression information, and a whole lot more. And because of, as we've said, the way that natural language processing works, once you've designed your query, it can be systematic and comprehensive, whether you're looking across one document, 10 documents, 1 million or 100 million. So we wanted to kind of think about some of the different perspectives and different challenges that, I mean, there's a spectrum really across healthcare and life science. If we look at the left-hand box here, thinking about the patient perspective, really, as a healthcare professional sitting across the desk from a patient, a pediatric patient or an adult, the key concern is, what is the diagnosis for my patient? So particularly with rare diseases, that healthcare professional may have never seen a presentation like this, and they need to get the best understanding of the range of phenotypes and then be able to compare that with a broad view of all diseases in order to rule in or rule out possible diagnoses. So, for example, they might be sitting with a patient with heart murmurs, tall, slender build, disproportionately long arms, legs, fingers, and they'd want to be able to combine that information together, match it, and enable, therefore, the suggestion of the disease being Marfan syndrome. So that's very much at the individual basis. When we think in the middle box here, broadening across a particular rare disease, you might want to say, okay, we're thinking about this rare disease, we're looking at, for example, Hunter syndrome. And you want to know, okay, across all my patients with Hunter syndrome that I may see, what's the prognosis for each individual across that group of diseases, that group of patients with the same rare disease? And what's the appropriate treatment for those? So here again, you need the landscape of information around the natural history of that rare disease. You need to pull that in from abstracts, from full-text papers, and again from EMRs, electronic medical records, as well as external, in order to get that landscape to think about how do we treat patients with Hunters depending on their particular presentation. And then the box on the right is really taking another step back and looking across the landscape of all rare diseases to help with that drug development, drug discovery pipeline. So here the question is how do I find existing drugs that might treat a rare disease? Obviously, any kind of indication expansion or drug repurposing means you're taking a drug that's already been through clinical trials and repurposing for those rare diseases, so that really can speed up to fill that gap of unmet need. And again, being able to pull out the associations between rare diseases and their genes, pathways, drugs, tissues, and more provides that network for drug repurposing. And again, enables those novel connections to be made. So we're now going to talk in more detail about 3 different use cases. This is going to talk about a healthcare use case from the University of Iowa. And I'm going to address 2 use cases. Again, think about those perspectives we were just talking about. So very much focusing on a patient, a particular rare disease area, and then a group of rare diseases. So I'll hand over to Liz.
Thanks, Jane. So Jane just introduced us to 3 different perspectives. Let's start our use case with the patient perspective, with the University of Iowa and how they use the human phenotype ontology to more effectively diagnose their pediatric population with rare disease. So first, let's set the stage of how generally NLP can help with EHR records. So if you look at this visual, this is a good example of the types of information that we can extract from a patient record. So as you can see from the examples we have, there's the text that is abstracted from many different types of documents. And then that information can be normalized. Like, for example, all the dates shown in yellow on the right. They can be abstracted in varied forms and then normalized into one format. Or look to diseases, the light blue section. So DM can be normalized to diabetes, and even a SNOMED code can be assigned. So there are many different types of clinical documentation that have important patient information trapped within unstructured text, which is actually captured. What is actually captured is it's up to you and your institution's priorities. So let's take a moment to look at the human phenotype ontology, or HPO. So the phenotypic information that we are interested in and want to extract from the medical record is available as part of the HPO. This is a formal ontology. It was created for medically relevant abnormal human phenotypes. So this includes things like macrocephaly, which is an abnormally large head, seizures, structured heart defects, etc. What it doesn't include are phenotypes considered normal or found within a large population, just variants. So this ontology is broken down into 5 sub-ontologies, of which phenotypic abnormality is the largest and the one we are interested in for this use case. The University of Iowa does significant testing on their pediatric population to ensure that if there is a presence of a rare disease, it is caught as early as possible to ensure that there's proper clinical care for the child, especially since sometimes a genetic defect could actually be life-threatening. So one of the issues is they are experiencing many test results that are inconclusive. They find that over 40% of their results are variants with unclear clinical significance. So therefore, the more phenotypic information that's available from the records is the more diagnostic yields come from the actual test. So prior, they did this manually, taking about 21 minutes per patient. They found on average 29 phenotypic concepts from the clinical notes. So utilizing Lingomatics NLP algorithms, we are finding over 4 times as many phenotypical concepts, so 131. And we're running 6 seconds per patient as opposed to the 21 minutes manually. Early assessments were found to be about 90% accurate. So assessment of the gold standards and assessing the accuracy of those and subsequent extractions has helped us understand the ability for NLP to improve this otherwise manually intensive process. And now back to Jane for more use cases.
Thank you. So I'm now going to talk through some work that Shire and now Takeda are doing, looking for genotype-phenotype associations for a particular rare disease, Hunter syndrome. So Hunter syndrome, or mucopolysaccharidosis 2, is a rare X-linked recessive disorder, and its deficiency of a particular lysosomal enzyme, iduronate 2-sulfatase, or IDS. And it leads to a progressive accumulation of a particular compound, a sugar compound, glycosamine glucans, throughout the body. So the trouble is with the accumulation of this is you get a set of signs and symptoms because of it. So bone deformities, respiratory infections, cardiomyopathy, particularly neurocognitive impairment and reduced lifespan. And some of these symptoms are improved with enzyme replacement therapy. But the spectrum of clinical severity can vary quite largely from mild to severe, with the main difference being progressive development of neurodegeneration and cognitive impairment in the severe form. So the FDA approved a particular enzyme replacement therapy from Shire called Elaprase in 2006. And that same medicine was approved by the European Medicines Agency in 2007. And in approving Elaprase, this enzyme replacement therapy, the EMA required Shire to sponsor and maintain a patient registry. So thinking a bit more about patient registries for rare disease. So a patient registry or survey can provide additional information about the course of disease, response to treatment, and safety. Now, when we think back to developing treatments for rare diseases, there are unique challenges because it's a rare disease. By definition, the patient pool is limited. So even clinical trials are very small and patient populations very small. So, for example, the key clinical trial for Elaprase was only 96 patients. And that was actually the largest trial ever for this kind of a disease at the time. So when you think about being able to understand the efficacy, the effectiveness, the safety of a particular treatment, you really need a registry set up to really follow these patients. And that's exactly what the Hunter Outcomes Survey is. It's a global, multi-center, long-term observational study. It's supported by Takeda, previously Shire, and it covers data for more than 1,000 patients across 30 different countries. So critically, where does natural language processing fit in? So as part of the many different reports within this registry, natural language processing enables adding broad literature context to increase the understanding of Hunter syndrome. So if you look at this example case report form, you can see on the bottom right, there's a series of boxes around particularly kind of mutation type information and phenotypes. And what we can provide with natural language processing is better annotation for that mutation. And if there's information on any literature or published phenotypes. So we've built a workflow, an automated workflow to extract mutations for that one particular gene, the IDS gene, and associated phenotypes from scientific literature. And then we can use that to enhance the information in the HOS report. So I'm not going to go into details. As we said, Shri will be giving you a demo. But in essence, the workflow is to start with abstracts, free text, pull out the right information for IDS, Hunter syndrome, mucopolysaccharidosis 2, find all the different forms, and then build a corpus, and then be able to start drilling in to connect mutations for the IDS gene with disease severity. So this is just a snapshot of some of the information you can find. And you can see that we're pulling out what the document is, whether the severity has been annotated in the text as mild, severe, for example, and then critically being able to pull out the different mutations, not just find them highlighted in pink in the snippets of text, but also normalize them. So again, making it very easy to understand, consume, and analyze. This can be pulled out from tables, as shown in this snippet of text here, or from free text, as you can see with this example. So again, this is using natural language processing to find the specific mutations around for this one gene and connect those with disease severity. And really, this has been a very effective extraction of valuable data for the Hunter Outcome Survey Disease Registry. We've been able to identify many observations, and particularly from about 460 full-text articles, find approximately 380 unique mutations. And for example, before text mining for this one particular position, R468, we've been able to increase the number of known mutations from 2 to 6. So again, really bringing benefit to the understanding of the natural history of this disease. And again, being able to link those mutations to the phenotype for that neurocognitive development for mild, severe, intermediate, and so forth. So overall, to summarize, we've been able to pull out the right information to annotate those Hunter Outcome Survey case report forms. The manual alternative would be slow and tedious and limited. We've been able to capture genotype-phenotype associations from literature, and that really feeds into the current understanding of Hunter syndrome, for example, risk factors for that severe phenotype. Moving on to our last case study, we're thinking back to those kind of 3 different types of challenges, the specific patient, the specific rare disease, and now thinking in a much broader picture, being able to look across very large volumes of literature, scientific literature, databases, and so forth, to be able to connect information across rare diseases with drugs, pathways, causal genes, and so forth, and look at the linkages in order to think about repurposing. So this is work done by Sanofi, where they wanted to be able to build network views of these integrated data for rare diseases. So obviously, if you're familiar with this kind of network approach, it's very popular for repurposing, for target new indication searches. It gives you a system-level view of a range of disease. It enables researchers to come in, look for commonalities between pathways, for example, new targets, disease gene relationships, and patterns in the networks may uncover underlying biomedical characteristics and therefore lead to possible solutions. So Sanofi developed an internal rare genetic diseases knowledge base, RGDKB, populated with the entire rare disease collection from Orphanet and Omin, with information added from literature using NLP to pull out causal gene associations. And the kind of information they can connect using natural language processing is not just direct from a target to a disease, as shown in the schematic here, but going through a whole suite of linkages. So these indirect relations allow networks to be built. So thinking from target to pathway, and then pathway to disease, target to cell type, and then cell type to disease. And so you can make these indirect associations and connections. And what they were able to do is present this as a visualization that they call RareView, which makes it very easy for end users to navigate the data that they've extracted from these big data sources, Orphanet, OMIM, scientific literature, and really review and understand the connections between diseases, genes, pathways, and therefore be able to potentially identify drugs that are targeting specific disease-causing genes and pathways, and therefore understand what could be potential for repurposing. And the quote at the bottom comes from the Sanofi team. Text mining with network approaches allows to quickly identify new indication opportunities from target disease pairs that have various commonalities based on broad scientific, medical, and strategic values. So it's a really powerful way to think about how to start dealing with some of that unmet need within the rare disease. So I'm now going to hand over to Shri to give a demonstration on how NLP can really provide rare disease insights.
Thank you, Jane. I'll go ahead and share my screen. Just bear with me here. So for my portion of the demo, I'll be showing you a few result sets from our Lingomatics NLP platform that emphasize what Jane and Liz have been talking about so far. The first result set here is looking at extracting phenotype information from de-identified electronic medical records. So the default output format is an HTML table that you see here, and it shows up in your browser tab. But you also have a variety of other options for your output formats. Here we are looking at document level results because these are de-identified records, but if you have patient information, you can definitely view the results at a patient level. In this result set, we are extracting phenotype information, a broader phenotype term, and both of these are coming from the human phenotype ontology that Liz talked about earlier, as well as a diseases ontology, which is a combination of the NCI and MESH ontologies. We can also distinguish whether a phenotype is for the patient or a family member. So here again, we've used our family terms ontology to pick up the different terms. So in addition to extracting the phenotypes as well as the patient or family terms, we are also extracting a clinical course, which is looking for terms such as acute, chronic, etc., and clinical modifier, which is looking for terms such as negative, mild, moderate, severe, and so on. Note that these two columns here are optional, so the phenotypes and the family terms will still be extracted regardless of whether there is a mention of a clinical course or a clinical modifier. So both of these, the clinical course and the clinical modifier, as Liz mentioned, are coming from within the human phenotype ontology. The leftmost color-coded columns here are specific to your query, and you have full control over the number and order of these columns. The last few columns to the right are what we call as evidence columns, and by default, they appear in every single result set that's generated from our NLP platform. One of the more important aspects of our NLP platform is that we're not just returning a handful of results, but we're also pointing you to where in the document these results are occurring from. So let's look at a sample document here. So here it says, has diabetes in brother and sister. So we've tagged brother and sister, and we've extracted the terms for diabetes here. Heart disease in both sides of the family. So for family, you have a broader term, cardiovascular diseases, and then the heart diseases for the phenotypic abnormality. And then it says father had brain cancer. So you have father here picked out, and then the neoplasm for the general term and brain neoplasms. It also says he denies any bleeding disorders. So it's tagged as negative for abnormal bleeding in the patient. Now, if you click on one of the highlighted text, you will actually be taken to a cached version of the de-identified medical record, where you can see the different hits highlighted here as well. So you're able to quickly get more context around these results. In our NLP platform, you have the ability to combine searches or queries into what we call a multi-query. The last column here tells us exactly which of those queries this particular result is picked from. So if I hover over the number here, you can actually see the query name that was used to generate this result. So this is looking at it from a healthcare perspective and electronic medical records. But moving along to pharma, along the lines of what Jane was talking about, we're looking at extracting gene-disease mutation type relationships. The next few results that I'll be showing you are coming from the last 5 years of Medline abstracts. Here we are looking at mutations associated with a gene. If we expand on one of these, for example, you can see that irrespective of how the mutations are written within these documents, they're all normalized to a preferred term, like you see here. So being able to do these kind of normalizations enables us to cluster the results like you see here. So you can expand on any of these blue arrows to see more results. And having a structured table of results like these with the values normalized can be really useful for downstream processing. Like I mentioned before, the first two columns here are specific to this particular query and result set, but then you have our usual evidence columns here as well. Again, if you click on one of these highlighted texts, you will be taken to a cached copy of the actual Medline abstract where that particular result is coming from. You can see the different results being highlighted here as well. So moving on now, this next set of results builds on the previous one. The results here are presented in what we call a chart format. So here the main feature of this format is that in addition to getting this table of results, which is similar to what we saw earlier, we also have a whole bunch of facets on the left, which we can use to filter these results further. So in this result set, in addition to extracting genes and mutations, we are also extracting the disease mentions that are associated with these genetic mutations. For example, if you want to look at mutations associated with a specific gene, you're now able to click on that gene, and then your results gets filtered. And let's say you want to further drill down your results and look at just a specific disease. So the results are filtered, and this table is now updated with the filtered results. Here we are also extracting whether there is a mere sentence co-occurrence between the disease and the gene, or if there is a direct verbal relationship. For example, if I click on the sentence relationship type, you can see that the diseases and the genetic mutations are here merely occurring within the same sentence. They're not necessarily connected by a verb. Whereas if I click on verbal, for example, you can see that there is an actual verb that's connecting the disease to the genetic mutations. So if you're doing more of an exploratory search, you may be interested in getting high recall, and you may just want to do a sentence co-occurrence. But if you're looking for a more precise set of results, then you can go for the verbal relationship type. You can click on the export option here. So you will be able to export this entire results table as a TSV file. And there's also an export column in the end here. You're able to unselect some of these results if you think that they're not needed in your final result set. You're able to do some level of curation at this point. So the chart format itself has a lot of options for viewing as well as exporting results. Moving on to the last set of results that I have to show you. Here again, we are looking at results extracted from the last 5 years of Medline. Here, in addition to extracting the mutation terms, we're also telling you whether it's a general mutation or a point mutation. In addition, we're also extracting the severity of the phenotype, mild, moderate, severe, and so on, and patient age as well as last author. So for patient age here, we are using our age ontology. Again here, to what Jane was mentioning about normalizations, the age is like greater than 40 years will be normalized to a standard format like this. And the same holds true for other numbers and age intervals as well. In addition to extracting concepts using your queries from these documents, you're also able to pull in metadata information as part of your result set if you so desire. So here we have pulled out the last author information. So as long as it's mentioned within the text, you can also extract other information such as patient number, study location, and so on. And you're also able to pull in metadata into your result set like this. So to conclude, I just want to note that the platform has a lot of flexibility, and it can present results in a very easily consumable format. I hope that this demo was helpful to you in understanding the capabilities of our NLP platform. With that, I'm going to hand it back to Jane for closing comments. Thank you.
Wonderful, thanks very much. So I hope you can see our summary slide. So I hope that's been of interest to you. We've walked you through the power of natural language processing text analytics and where it can bring value to on the healthcare side, on the pharma side, particularly focusing on rare diseases, being able to pull out in a rapid, systematic, comprehensive way the insights that are needed in unstructured text. And the little schematic on the right-hand side here of the iceberg is just to remind us that, particularly for sort of genetics, genomics, there is a huge amount of information in structured databases. But for all of the use cases we've talked about, there's just so much more information that's of real value that's buried in the unstructured data. And in fact, you know, the standard metric is something like 80% of all data relevant for biopharma, biomedical applications is actually in unstructured data. So if you want to really get your arms around that, then using an AI tool such as natural language processing really provides that. So specifically, what have we talked through? So we've talked about how the information associated with genotype-phenotype associations, disease associations, is essential to build an understanding of rare diseases, either at that patient diagnosis level, either thinking about registries and understanding at a rare disease level, or really broad across many rare diseases, thinking about drug repurposing. And critical information is locked in unstructured documents, such as abstracts, full text papers, electronic medical records, and manual curation, as we all know, is slow and tedious and limited and can be error prone. So natural language processing can transform the wealth of both internal documents, external data, into high-value actual insights. So if there's one take-home message, it's to go away with thinking about how NLP can provide a rapid, comprehensive access to enhance gene disease and genotype-phenotype associations to provide decision support for rare diseases.